All right, well, thank you all for coming. Thank you for joining us for our third annual long-term memory encoding and Connectome decoding meetup. We're working on the name. In a moment, I'm going to announce the winners of this year's Aspirational Neuroscience Awards, and then we can move on to the main event, which is the panel discussion. We have some really fantastic neuroscientists that are going to discuss what would it take to decode a non-trivial memory from a Connectome. I think it's going to be super interesting. But before I go there, I want to give kind of an overview of what we're trying to do. So a little over 15 years ago, a small set of neuroscientists were arguing that electron microscopy should be automated in order to allow for the dense mapping of neural circuits. So, for example, this 2009 paper from Sebastian Sung said this in the abstract. He said, another method, this is before connectomics, essentially, or the most recent round of connectomics. Another method will be to find the connectome of a dense map of connections in a single species and infer the functional properties of those neurons through a computational analysis of the connectivity. For the latter method, the most exciting prospect would be to decode the memories that are hypothesized to be stored in the connectome. And so right from the beginning of this recent round of EM connectomics was the idea that an ultimate test of connectomics would be whether you can really decode the algorithm, whether you can decode memory and learn function from the connectome. And the intervening years, let's say 15 years, there's been a technological revolution. Hopefully people caught the connectomics talks earlier today in the Connectomics 1 Nano Symposium. There's been a revolution in volume electron microscopy such that we now do have access to increasingly large and accurate connectomes across a wide range of model organisms. For example, we recently buffalo central nervous system. I thankfully had a small part in that project. Other labs like Allen Institute and their collaborators have produced this wonderful microns connectome spanning several visual areas and complete with calcium imaging that on those neurons. Additional labs have mapped connectomes from other cortical areas and have begun to map the hippocampus. Whole zebrafish connectome is going to come online soon, again with paired calcium imaging, and the songbird, which has long been a model organism for studying the basal ganglia, is also having its connectome mapped. In fact, these electron microscopy volumes are they're likely to be eclipsed in the next few years. In terms of volume, by light microscopy techniques like Glycon that use expansion microscopy and pan-protein staining that have been shown to be able to also produce connectomes. And in parallel, there has been a revolution in the genetic tools that can tag and manipulate engram cells that activate when a memory is recalled. Tools like eGrasp that allow tagging of synapses on engram cells, and even the manipulation of synapses through Cali and others. And so, given all these advances, we thought these advances in connectomics and memory engram manipulation, we thought it was time to revisit the bold ambition that kicked off, one of the bold ambitions that kicked off connectomics revolution in the first place. Can we decode a non-trivial memory from a static connectome? And I think it's probably agreed that that has not occurred yet. It's not a really clearly defined goal. Maybe we can discuss that tonight. But I think it is an achievable goal. And so the Aspirational Neuroscience Outreach Project is its goal is to bring together researchers in connectomics and researchers in memory engrams in order to design concrete experiments with the explicit goal of decoding memories from a static map of synaptic connectivity. And one of the important things to take home from that is that such an experiment would really put to the ultimate test the theories of memory. We want something that really is, for this particular system, for this particular type of memory, is this decoding challenge would ultimately say whether that is a true theory or whether it is not. Okay, so to that end, we have thankfully got a generous donor that is very enthusiastic about neuroscience, and we have put forward a Memory Decoding Challenge Prize with the goal to help motivate such collaborations and to spark discussion about this. It's a $100,000 prize for the first neuroscience research team who can demonstrate the decoding of a non-trivial memory or other learned function from a static connectome. Now, defining exactly what it would constitute a non-trivial memory is something that I hope we can discuss in the panel tonight. In essence, though, it should constitute a crystal clear demonstration that a neuroscience model of how memories are encoded in the structure of neural circuits is actually correct. And I want to make clear that I don't see this as some impossibly distant goal. I see many research teams at SfN presenting research that are actively pursuing projects that could constitute such a demonstration. So here's some concrete examples. I wanted to throw some concrete examples in just to help people kind of understand the scale that we're talking about. So for instance, one decoding project that Jorgen Kornfeld has been discussing for a while and is hotly pursuing is decoding the learned song from the connectome of the songbird HBC to RA nucleon. Decoding the rudiments of a spatial map in the hippocampal system would certainly constitute decoding a non-trivial memory. Decoding a set of specific fear memories in the amygdala might constitute it. Decoding receptive field properties of many visual cortical neurons, or higher order cortical visual neurons, and many others. And hopefully we can discuss this in the panel. Now, because it's likely to take many years for any team to reach that milestone of decoding a non-trivial memory, we're giving 4 $25,000 Aspirational Neurosciences Awards, hopefully each year. We're trying to do this, for research that seems to be on the path toward that ultimate goal, or is contributing to our understanding of memory in a deep way. And these are just some of the winners in past years. We also run a bi-weekly journal club with a large but unfortunately not too active email discussion group. I want to invite anybody that really wants to dig into the literature, because we need help. We need experts to keep on top of the latest research. We frankly would also like people with deep expertise in the field to help with nominating the right types of papers and judging those. Okay, so let's move on to this year's Aspirational Neuroscience Awards. Our website has a list of papers that have been nominated for Aspirational Neuroscience Awards. This is a collective thing. It's not like they were nominated this year and they didn't win, so they can't win the next year. This is a collection of papers that are all have been nominated. So any of these can potentially win next year. So our website has a list of papers that have that our team has nominated for Aspirational Neuroscience Awards. They have been sorted roughly into different categories, just to give you an idea of what we're looking at. Correlation among synapses with shared history, dendritic integration and neuronal modeling, fear memory in the amygdala, a whole bunch of hippocampus work that you can imagine, labeling and photo erasure of synaptic ensembles, molecular mechanisms of learning, synaptic structure function correlations. The synaptic structure function correlations is really the name of the game in decoding a memory from a static connectome. But all of this research that is really fundamental research about how what the different learning rules are we should be on top of if we're looking for what can be done in connectomics. I missed one, and visual receptive field. Okay, so without further ado, these were the 12 that were nominated this year. Again, the other ones are still in the running. I didn't decide who would win. There is. I could make arguments that each one of these is the ultimate paper. So if anybody is here that is not winning an award here, please accept my apology. But there were we had to choose one. Okay, and here are the winners. Co et al. 2025. Dorkenwald et al. 2022. Lampeliner et al. 2024. And Ding et al. 2025. Again, I want to say there are some absolutely earth-shattering, fantastic papers that didn't win, and this should not be. We had a lot of discussion about it, and it was along the lines of, yes, this is earth-shattering, but is it on the critical path? Yes, this was earth-shattering, but it was a little too many years ago. Things like that. It should not be. Everybody on this list was just fantastic. Let me give a very brief overview about the ones that won. So the paper, Systems Consolidation Reorganizes Hippocampal Engram Circuitry by Co et al. Showed that shifts in episodic memory precision from specific to general, which is typically associated with cortical consolidation, may actually be partly explainable by synaptic reorganization in the hippocampus itself. They used a combination of engram cell tagging and eGRASP synapse labeling to track the synaptic modifications underlying this GIST memory formation. So one of the things that we found was very impressive was tracking the change of a memory through tracking its synaptic changes. Okay, another one is this paper by Dorkenwald et al. 2022. Dorkenwald will be on the panel discussion. The paper, Binary and Analog Variation of Synapses Between Cortical Pyramidal Neurons. They traced and proofread 1,960 synaptic connections among 334 pyramidal neurons in a TEM data set, covering layer 2-3 of mouse visual cortex. They quantified the size and thus the functional strength of each of these synapses. Plotting a histogram of the synapse sizes showed that instead of the expected log normal distribution, a highly skewed distribution that was better fit by a mixture of two log normal distributions was seen. Then they analyzed a subset containing 320 of these synapses from 160 matched pairs, two synapses that shared the same pre- and postsynaptic cells, such that they had the same learning history. And plotting a histogram of the geometric means of each of these pairs showed that an even clearer bimodal distribution, which the authors showed could be modeled by assuming that each pair had a binary value, either small or large, with an uncorrelated analog variable on top. So this is evidence that Hebb-style learning rule with only two synaptic strengths, small versus large. This is this is kind of cutting edge stuff. It showed the real strength of having a large, accurately traced connectome. That was one of the key things. This is kind of a result that is showing off the strength of connectomes. So, okay. Another paper, Connectome constrained networks predict neural activity across the fly visual system by Lapalainen et al. They showed a novel way to predict function from a static map of synaptic connectivity, even when many dynamic parameters of the neurons are not known, and synapses are not known. They generated a differentiable model of the fly visual system based on a full EM connectivity. And this allowed the unknown dynamic parameters to be tuned by gradient descent with the goal of matching a known motion selectivity of a subset of the neurons. We thought that this novel method showed how the considerable constraints of a connectome could be used to leverage even if not all the dynamics was known. And I've heard even in talks around here that people are thinking about applying this to the Micron's data set and other data sets because this is, you always have some dynamic factors in neurons that you don't have when you have the connectome. So this shows a path forward for these. So finally, the paper Functional Connectomics reveals general wiring rule in mouse visual cortex by Ding et al. At some level, this paper had to win. It's a very the Microns project is perhaps the most ambitious connectomics project to date. It acquired a cubic millimeter volume of electron microscopy data spanning multiple primary and higher order visual areas. Overlapping regions were calcium imaged in the awake behaving mouse while watching hours of specially designed video stimuli. The paper analyzed a subset of neurons having both high quality calcium recordings and proofread connectome tracings. And this allowed the authors to ask questions like, are the neurons that respond to similar visual features more likely to be synaptically connected or not? And the authors employed some really novel analysis techniques, or at least we did not see them in other papers at this level. They quantified the distance each axon travels in close proximity to a dendrite. And this allowed them to normalize the actual synaptic density between any two cells. It's kind of like, they're not just saying these two cells are connected, they're asking first the question, were they close enough to be connected? And then did they have a synaptic connection? A much stronger, tighter statistical question. The authors also employed a digital twin to precisely quantify each cell's receptive field location and its feature tuning. And intriguingly, they showed that cells with similar feature tuning have higher than expected synaptic interconnectivity, a result that other groups had seen before. But they also showed that cells with similar location tuning show lower than expected synaptic interconnectivity. And I believe that has some serious implications for our understanding of how learning happens in the visual system and other sensory systems. So in any case, these are the papers that were awarded this year, and we should give them another round of applause. Okay, so now we are going to go to a panel discussion that is moderated by the amazing Paul Middlebrooks, and take it over.